CRISPR gets all the glory for cutting. We talk about it as molecular scissors, a tool that can find one exact sequence in a living cell and snip it. And that is true. But scissors are only safe because you can put them down. Imagine a pear that, once opened, never closed again. That is not a tool anymore. That is a hazard loose inside the cell. So here is a question we almost never ask about CRISPR. Not how do you make it cut? How do you make it stop? Today we are going to follow a team that answered that question in a way nobody had done before. They did not search for an off switch. They designed one from scratch, using artificial intelligence, for a version of CRISPR that had never had a break in the first place. Let us get our bearings. CRISPR started as a bacterial immune system. A bacterium keeps little snippets of genetic memory from viruses that attacked it. It loads one of those snippets, a short guide, into a cutting protein. The guide is a sequence. The protein carries it around and compares it to whatever it bumps into. When it finds a match, it cuts. The famous version, Cas9, cuts DNA. That is the one used to edit genes, to fix mutations, to rewrite the code of life. But Cas9 is not the only member of the family. And the one at the center of our story does something different. It does not touch DNA at all. It hunts RNA. This is Cas13. RNA is the working copy, the message a cell reads to build proteins. Cas13 takes a guide and goes looking for one specific message. When it finds the matching RNA, it cuts it, using a part of itself called the heparin domain. Remember that name. Heparin is the blade. But Cas13 has a strange and dangerous habit. Once it makes that first cut and switches on, it does not switch back off. It keeps going. It starts slicing nearby RNA at random, messages it was never aimed at. Scientists call this collateral cleavage. In a test tube, that runaway behavior is actually useful, and it powers some clever diagnostic tests. But inside a living cell, an enzyme that cuts its target and then shreds everything around it is exactly the kind of thing you want to be able to shut down on command. Every powerful tool in biology eventually needs a break, not because the tool is bad, but because timing is everything. You may want CRISPR active for 1 hour and silent for the next. You may want it on in one type of cell and off in another. You may simply want a safety stop, a way to call the enzyme back before it does damage you did not intend. For gene editing to be safe enough for medicine, that control cannot be a luxury. It has to be part of the toolkit, which raises the obvious problem. Where do you get a reliable, specific off switch for an enzyme as aggressive as Cas13? It turns out nature already invented breaks for CRISPR. Bacteria and the viruses that infect them have been at war for billions of years, and the viruses evolved small proteins that jam CRISPR so they can survive. We call them anti-CRISPR proteins. They are real, and some are remarkably good. But there is a catch. Finding them is painfully slow. You essentially have to go hunting through the genomes of viruses and hope to stumble onto one, then prove in the lab that it works. Over an entire decade of this, researchers validated only about 118 of them. And for Cas13 specifically, the RNA hunter we just met, there was not a single confirmed natural inhibitor. The break we needed most simply did not exist in the catalog. So a team at Monash University and the Bio21 Institute in Melbourne tried something audacious. Instead of searching nature for a break that might not be there, they decided to build one. Not modify an existing protein. Invent a brand new one, a shape that had never existed in any living thing, custom fit to grab onto Cas13 and hold it shut. A few years ago, that sentence would have sounded like fantasy. Designing a working protein from nothing means choosing, atom by atom, a structure that will actually fold up correctly and do a job. The number of possible proteins is astronomically large, but this is exactly where artificial intelligence has changed the game. The first tool they used is called RFdiffusion. You can think of it as an AI sculptor. It starts from random noise, a meaningless cloud, and step by step it denoises that cloud into a coherent protein backbone, the structural skeleton of a new molecule. Crucially, you can tell it what the backbone has to do. Here, the instruction was simple. Generate a shape that fits snugly against Cas13, right over its business end. This is the same family of AI as the image generators that turn noise into pictures. Except instead of a picture, it is producing a three-dimensional structure that has to obey the laws of physics and chemistry. Noise goes in. A candidate break comes out. A backbone is only a shape. To turn it into a real protein, you need a sequence, the exact chain of amino acids that will fold into that shape on its own. That is the second AI, called ProteinMPNN. Give it the backbone, and it writes the sequence most likely to fold there and stay there. And the target was precise. Not just Cas13 in general, but the heparin domain, the exact part that does the cutting. The strategy is simple to say and hard to do. Plug the blade. The designed proteins, which the team calls Ikers, are shaped to settle into that active site like a key broken off in a lock. With the site occupied, Cas13 cannot reach the RNA. The scissors are jammed shut. Designing something on a computer is one thing. Proving it works is another. So here is the part that genuinely surprised people. From picking the target to holding a working inhibitor in hand took about 8 weeks. Compare that to a decade of searching that produced no Cas13 inhibitor at all. And these were not just pretty simulations. The designed breaks were made for real and tested. They shut Cas13 down inside living bacterial cells and inside human cells. A protein that did not exist 2 months earlier was now reliably switching off one of biology's sharpest tools, on demand. Step back and look at what changed. For years, our control over CRISPR was limited to the breaks that evolution happened to leave lying around. If nature had not made one for your enzyme, you were stuck. That constraint is gone. We can now design new inhibitors on demand for whichever CRISPR system we need to control in weeks. Let us be honest about what this is and is not. This is a proof of concept for a new kind of tool, a way to make CRISPR safer and switchable. It is not a treatment sitting in a clinic, but the bigger story is the method itself. A safer, controllable CRISPR is no longer something we have to wait for nature to provide. It has become a design problem, and the designs are already working. That is the shift worth remembering. We used to search for the off switch. Now we draw it. This is Paperscape. The paper is linked below. See you in the next one.